Welcome to our second webinar in the Frontier and Healthcare and Information Technology Keynote Series. Today, we are really honored to welcome Dr. Roy Parker, whose presentation is entitled RNA Mis-Regulation in Neurodegenerative Diseases. Dr. Roy Parker received his PhD from the University of California, San Francisco, after a postdoctoral work at the University of Massachusetts, Worcester. He became a faculty member whose research focuses on the translation, localization, and degradation of eukaryotic RNA, how cells regulate different steps in the process to modulate gene expression, and how alteration in RNA regulation lead to human disease. Dr. Parker has served on and chaired on NIH study sections, co-organized meetings and conferences, and has been on the editorial board of various premier journals. He is currently at the Chuck Leonard End of Chair of Biochemistry and Distinguished Professor at the University of Colorado Boulder. He is the Executive Director of the BioFrontiers Institute, an elected fellow of the American Academy of Arts and Science, an elected member of the National Academy of Science, and an investigator with the Howard Hughes Medical Institute. Without further ado, Dr. Parker, the floor is yours.
Thank you, Elias, for that nice introduction, and thank you all for attending. What I'd like to do today is tell you about some of our work in the lab and about how it relates to neurodegenerative diseases. And to begin, I'm going to share my slides with you. Today, what I want to do is, my lab has had a long-standing interest in understanding RNA and how cells regulate RNA molecules and how that connects to human diseases. And in the last couple of years, we've really shifted our focus to start studying the connections of RNA physiology and some neurodegenerative diseases. And what I want to do in today's talk is start by talking a little bit about the problem of dementia, and then emphasize how dementia is frequently connected to a particular protein called tau and how that forms protein aggregates in cells. And then I want to show some of our recent work showing that tau aggregates in cells and in neurons actually contain RNAs, particularly RNAs that are involved in what's called pre-mRNA splicing. And then some data that we have that this might be part of the pathology of tau aggregation in various diseases by altering the process of pre-mRNA splicing and how we're trying to address that further. So that's kind of the overview of the talk. So to start, dementia is really a huge problem in the world, and it refers to a number of different symptoms, really, such as memory loss, difficulty completing familiar tasks, being disoriented. And dementia, is both a huge cause of death around the world. In the US, it's 6.2 million cases, sixth leading cause of death. And it's also extremely expensive to treat dementia because people can persist for several years with really under dysfunctional states. And we're all familiar with this. I think probably everyone has some individual in their family who's suffered through dementia at some stage. And there's a number of different diseases. Alzheimer's is by far the largest, about 50% of dementia, but there are other diseases which also make up about 50% of other kinds of dementia diseases. And one of the main characteristics of all these diseases is what's referred to as neurodegeneration. And really what neurodegeneration is death and loss of neurons. And so this is just showing brains from a normal individual, an individual who has Alzheimer's disease. And if you look at the structure of the brain, what you can see is that in the individual with Alzheimer's, a fair amount of the brain has been lost. And this is really because of the death of neurons and the shrinking of the structure of the brain tissue. And so then a huge question becomes, why do these neurons die? And what could we potentially do about that as we understand that process? So a common feature of many neurodegenerative diseases is the accumulation of aberrant protein aggregates. And this is just showing a picture of a brain slice from individual with Alzheimer's disease. And you can see that the neurons, which are normal here, don't stain with this particular protein called tau. But the diseased neurons actually accumulate large amounts of this protein, and it forms a aberrant structure, as I'll tell you about. And so, a fundamental question then is to understand what the aberrant protein aggregates are in neurodegenerative diseases, how they form, and how do they impact the cell, the neuron, to cause pathological processes. Now, a long time ago, when Alois Alzheimer was first describing the Alzheimer's disease, he identified in 1906 that neurons in what came to be called people with Alzheimer's disease had these aberrant structures in their neurons. They kind of formed these fibril structures, and he drew these pictures, from over 100 years ago. And then in 1986, it was discovered that these, what are called neurofibrillary tangles, are actually composed of an insoluble protein called tau. And tau makes these structures in neurons in Alzheimer's disease. And in fact, what's really striking about tau is if we go back to that pie chart of individuals with neurodegenerative diseases, with dementia diseases, if you organize them by disease type, you'll see only about 50% have Alzheimer's disease. But if you organize them by what kind of proteins aggregate and form aberrant structures in them, what we see is that almost 80% of the cases of dementia are what are called tauopathies and have these types of neurofibrillary tangles in them. And so this is why my lab has really started to focus on studying tau and how it goes bad, because it's really driving probably 80% of the cases of dementia in the world. Now, tau aggregates have been found in, I think that the current count is up to 27 different diseases. There are really two kinds of tauopathies that we worry about. One are what are called primary tauopathies, where tau is thought to be the driver of disease. And these are caused by a particular mutation in tau that leads to the degenerative diseases. So some of these are what are called frontal temporal dementia, where there are specific mutations in the tau gene in humans, which lead to that disease. And as I'll show you later in the talk, cortical basal degeneration, another form of neurodegenerative disease, where you have lots of these neurofibrillary tangles of tau, is also caused by specific mutations. And then there's also what are called secondary tauopathies, where tau aggregation is caused by something else, not a mutation in tau. And these include things like Alzheimer's disease. We know in Alzheimer's disease there are also these A beta plaques. And I think the emerging theme is that what A beta plaques do is they actually somehow trigger tau to start forming these neurofibrillary tangles. But other things can cause tau to form aggregates. The disease CTE, which is, we see in athletes who get repetitive head trauma, triggers tau aggregation somehow. And I want to point out too in red here, a disease called PEP. And this is actually a tauopathy which occurred after the 1918 flu. After the 1918 flu, there was a pandemic of a disease called encephalitis lethargica, where people had symptoms very similar to what you might imagine with long COVID, and that some of those individuals then developed a subsequent tauopathy and passed away. Measles, about 0.1% of people who get measles actually end up with a tauopathy about 10 to 15 years later. So what the important point I want to make here is that perturbations to individuals' health, including viral infections, can actually trigger a process in our bodies which somehow leads to this tau formation of tau aggregation and subsequent neuron death. And one of the, I think, things we should all be paying attention to is the possibility that this will actually occur with COVID as well. We know that other viral infections can, such as the 1918 flu or measles, can actually trigger a process which leads to tauopathies. And I think in a small subset of people, it won't be most people who experience COVID, a small subset may actually experience a tauopathy as well. And this is something that the people in the medical and neurodegenerative community should be keeping an eye on. There are programs being set up to look for this over the next 10 years. And the idea there is that what's common about many of these things that trigger tauopathies is that they increase neuroinflammation, and including viruses. And so if you have lots of neuroinflammation, either due to repetitive head trauma or viral infection, that can start this process which leads to tau aggregation. All right, now, why does tau form these aberrant structures? And I just want to remind people that, proteins normally in our bodies fold up into well-defined structures that have biological roles. And kind of a classic example, this would be the protein hemoglobin, which forms a very defined structure and carries oxygen in our blood. But tau is what's called an intrinsically disordered protein. So you can think of it like pretty much a squiggly protein, which doesn't form a well-defined structure. And because it's not in a well-defined structure, it can fold into different kinds of structures. And tau samples these different folding patterns at different times, kind of in a dynamic manner, dependent upon thermal fluctuations. And as a result of that, because some of these proteins that it folds, that it creates, can actually assemble into these larger structures that we see in cells that are called tau fibers or neurofibrillary tangles. And so the way that occurs is really in three general states. This would be a diagram of the amino acid sequence of tau, and it kind of can fold up into a structure where it interacts with itself. And then often these will then form a dimer with another similarly folded protein. And then this pair of tau proteins will stack on top of each other, like, as you can see here, like different layers. And that this forms what's called a very, very stable cross beta structure, which is hyperstable protein structure in our cells, and therefore can persist for a very long time and cause problems. And this process then is very similar to what we think of as a prion propagation mechanism. And so just to remind people, prions are diseases like mad cow disease, where if you have a protein that forms up into a particular very hyperstable structure, that protein structure can then interact with other molecules of the same protein and cause those other molecules to form the same type of structure shown here by the blue tau protein, now sitting down on top of this growing fiber to continue and extend the fiber. And so what this does then, if you think about this in the context of an individual, if you actually have a pair of neurons here, where a neuron actually starts to make tau aggregates, those aggregates can form what are called seeds, and those seeds get transferred to another neuron, and that triggers the process of more propagation of these fibers in the new neuron, which can then go on to even another neuron. And in this manner then, tau is really an insidious protein assembly, which can propagate from neuron to neuron to neuron in the brain and cause neuron death, and then therefore cause these kinds of neurodegenerative diseases which occur as a consequence of tau. Right, and so there's no evidence tau is ingestible like the mad cow context, but once it gets started in the brain, it can propagate within the brain. This is why it causes such debilitating disease. The tau, because tau has these different folds, because it doesn't have a defined structure. It can fold up into different kinds of structures in cells and form different types of fibers in different diseases. And so this is kind of interesting and a real challenge for those of us who are studying tau, because we don't know that this is a structure in some Alzheimer's disease patients, and this is a structure in some cortical basal degeneration patients. Are we going to be able to treat these with the same kinds of approaches, or do we actually have to understand the differences in each case and then define therapies that are unique to different types of tauopathies? So this is a challenge that we're going to have to face over the next few years. All right. So there's really, I think, a couple big questions that my lab has been trying to answer. How and where do aggregates form in neurons? Do they form in particular places? And then how do they affect cellular function? Why do these aggregates of tau actually trigger disease? And can we come to understand that? And if we can we then use that information to think about developing therapies? All right, so the work from my lab that I want to talk about really has two interesting, I think, or important discoveries. One is that I'm going to show you some data which convinces us that these tau aggregates in disease states are not just tau protein, they actually include RNA molecules. And in particular, they include RNAs that are called siRNAs or SNO-RNAs, and I'll talk about what they do in a second, for those of you who don't know. And then the other thing I want to talk about is tau aggregates actually form in a special place in cells, in a place called nuclear speckles. And these are interesting because they're actually involved in splicing. And I'll talk about how we think tau alters the splicing process. Now, this work is really all the work of a MD PhD student in the lab named Evan Lester, and I want to give him credit because he really led this project. And we actually interacted with Stan Prusner's lab. Stan is the individual who won the Nobel Prize for understanding prion proteins such as mad cow disease. And Stan's group has been really instrumental in helping us, particularly with some of the experiments with brains, because my lab doesn't have access to brains from patients who've had these diseases. All right, so why would we actually think that tau aggregates are composed of RNA and protein? Why did everybody thinks of these as just a protein fiber? And I just want to remind everybody that cells are actually full of RNA molecules. The ribosomes that translate RNAs are an RNA protein machine. The mRNA, which is kind of the key genetic instructions for making protein, of course, is an RNA. And tRNAs are the critical RNAs which actually decode the genetic instructions. But they're not just these RNAs, there are lots of other RNAs that have other roles, such as things involved in DNA synthesis, like telomerase RNA, or RNAs I want to talk about involved in splicing, referred to as small nuclear RNAs or siRNAs. So there's lots of RNAs in cells. And not only that, we actually knew that the tau protein is known to bind to RNA molecules. This is from people who've worked on this for over 20 years now. So tau binds RNA. There's lots of RNA in cells. And one of the things that my lab has worked on over the years are these structures called RNP granules, which are really ubiquitous in eukaryotic cells. So if you look inside of a cell, and you look at particular RNA binding proteins or RNAs, you can see that they're often organized into these structures. These are ones called stress granules, which exist in the cytoplasm of cells. There are other ones which exist in the nucleus. And in neurons, there are really interesting collections of RNAs and proteins which actually travel down dendrites and are involved in forming synapses. So we knew there were lots of these RNA granules. We knew tau-bound RNA. And so we thought maybe that tau might be bound to RNA in cells. And we were really intrigued by this paper a few years ago showing how tau fibers form in vitro. So I've showed you these are the kind of fibers which form in patients, that people had purified tau and shown that you could take tau in vitro and build these types of fibers from it if you just shaked it in a test tube for long enough. But one of the striking things that they observed was tau by itself could not do that. You had to add something else. And what worked the best was actually RNA. And so this is an experiment done by Martin Margita's lab, where he took tau protein, and if he adds RNA, he can actually make these tons of tau fibers as detected here in a gel. And if he keeps taking a little bit of this and adding it to a tube with RNA and tau, he'll make more and more and more and more. But if he leaves out the RNA, what happens is he very quickly stops making new tau fibers. And so this suggested to us, you know, maybe the ability to form these fibers in cells actually requires RNA molecules. And if you look in the structures of some of these are structures of tau fibers formed from patients where they're solved by using cryo-electron microscopy. And you often see these kinds of things in here. You can see this fuzzy region here that, and nobody really knows what this is. It's not, they haven't been able to solve the structure of that part of it, but it actually looks very much like it might be an RNA molecule. So maybe RNA is actually part of these fibers even in patients where they form the pathogenic disease. All right, so that's why we wanted to address this question. And so how do we go about this? You know, you can't obviously study this in people. That's too intrusive. But we can study this even in cancer cells that we can grow in the lab. And this is kind of the system we often use. We take a cancer cell line called HEC293 cells, and in that cell line, they express the tau protein, tagged with a fluorescent protein. And so that allows you to see tau when it forms these aggregates. And because tau is like a prion, if I take a brain extract from a mouse or from a human who had one of these diseases, and we transfect that, introduce those parts into the cell, they'll actually trigger the formation of new tau aggregates, just kind of by this prion templating mechanism. And if you do that from a mouse which does not have disease, then nothing happens. All right, so this gives us a way to actually study this process in a very simple system that we can manipulate in the lab. And I just want to point out that we actually see, you know, if you zoom in on a cell here, and the blue is the nucleus, it's showing you the DNA, you can actually see that you form these big tau aggregates that are out in the cytoplasm. But in the nucleus, you also form particular tau aggregates as well, and I'll talk a little bit more about those in a second. So what Evan did was then he basically lysed these cells and figured out a way to purify these tau aggregates. And this is just a picture of one that he's purified. And if you stain them for RNA, you can see that that tau aggregate stains very brightly for RNA molecules. And so these tau aggregates actually contain RNA. So then he took these purified assemblies and then he prepared libraries to sequence them. And he used DNA sequencing to sequence the RNA molecules that are in there, and then identified those types of RNAs that were present and how they were enriched. And what he saw, and this doesn't really, the details of this aren't important, this is just showing you the enrichment of certain RNAs and tau aggregates as compared to a control setting. And what you can see is that there's some of these RNAs which are red dots, which are quite highly enriched in the tau aggregates. And this is actually from a mouse, which has been genetically engineered to express a particular mutant form of tau, which caused disease in humans. But what I want to draw your attention to is that there's one class of RNAs which are very highly enriched in tau aggregates, and these are what are called snRNAs. And snRNA stands for small nuclear RNA. And what these RNAs typically do is they perform a process called pre-mRNA splicing. And just to remind everybody what pre-mRNA splicing is in human cells, when you turn a gene on and make the RNA, it contains regions that are called exons, and those are part of the gene which actually encode the protein. And then it has a region called an intron, which needs to be removed in order for the mRNA to function. you can think of this analogous to a commercial in a television show. And so the cell has a system where it recognizes the intron and then cuts it out and puts the RNA together, removing the intron from the RNA. And that's referred to as splicing. And these siRNAs are part of this process. And so these are things like termed U1 or U2, and they bind to the intron and orchestrate the chemical reactions which remove the intron from what's called the pre-mRNA to make them the real mRNA, which is then used by ribosomes to produce protein. All right. So, so there's a bunch of these siRNAs in these tau aggregates, and this is the first hint that made us think that maybe these tau aggregates are changing pre-mRNA splicing. I'll come back to that in a second. So tau aggregates contain these RNAs, and I think this is a really important discovery because it suggests to us that maybe tau aggregates forming in individuals actually form in a process that involves RNA molecules. And we're really entertaining two models at this time. One is that tau aggregates form just because of the protein, that they then can recruit RNAs to them to just stick there. I don't think this is likely because in the test tube, you require RNA to make these fibers. You can't do it without an RNA molecule. And so, so we're trying to test the other possibility that actually what happens is tau binds RNA molecules, and that's what these little cartoons are, cartoons of RNAs, and that then that RNA actually helps the formation of these higher order structures, which end up causing these fibrillar disease events. All right, so we're working on trying to test that. That's actually not an easy experiment to do, but I hope maybe in a year or so we can answer this question. But in the meantime, we've been interested in, I mentioned that there are these cytoplasmic tau aggregates, but there are also a second class which form in the nucleus. And we've been interested in what these nuclear aggregates are and where they actually form. So what Evan did was he did a number of experiments to examine if these nuclear tau aggregates overlap with other nuclear structures. And I'm just going to show you one of these where we actually stained for a nuclear structure called a nuclear speckle. And so that's this protein here, SRM2, that we detect with an antibody. And you can see that these nuclear speckles form a structure in the nucleus, which is kind of parsed around in these stringy or dot-like structures called speckles. Now, when we seed tau aggregation in these cell lines, we get these big cytoplasmic assemblies that form. But you also see these form in the nucleus as well, and they form right on top of where the nuclear speckles are. Right? And so if you merge them, you'll see that you get yellow speckles in all of these cells. All right. So what are nuclear speckles and why do we care? And one of the things that we, the scientific community, has learned in the last 10 to 20 years is that cells contain a large number of non-membrane bound assemblies. These are often referred to as biological condensates now, that concentrate proteins into some types of organelles, which allow those organelles to have functional properties that are different from the rest of the cell. And nuclear speckles are one of those types of biological condensates. And part of what my lab works on is trying to understand the properties of these assemblies as well. And if there's questions, we can talk about that. So the tau aggregates are forming in nuclear speckles. And what nuclear speckles are thought to do is there's thought to be sites where RNAs, when they're first produced, concentrate in the nucleus. So this would be the newly transcribed RNA molecules, and they actually concentrate the splicing machinery. And they're thought to have roles in both the production of RNA, maybe the control of splicing, and also the regulation of RNA export, although this is not very well understood, to be honest, and it's going to require some more work. So this has gotten us interested then. Okay, so how does tau interact with these processes of speckles? And but before we dug too deep into this, we looked in mice because this here we're studying the aggregation of tau in a cell line model, and this is true in an organism that actually has neurons. And so I just wanted to show you this work here. Here we're looking at, this is zooming in on the nucleus of a neuron in a mouse, which was expresses a pathogenic form of tau and develops disease. And you can see that this tau aggregate actually overlaps in the same place with a marker of a nuclear speckle. And so what this tells us is that this formation of tau in nuclear speckles is not something that's unique to a cancer cell in culture. It occurs in mouse models of disease, and it also occurs in patients. And there's lots of papers now showing that there's nuclear tau aggregates in patients who have these tauopathy diseases. This is actually not studied very much because the vast majority of the tau protein is out in the cytoplasm, but these nuclear tau aggregates could be very important in the disease process. All right, so while we were doing these experiments, we saw something completely unexpected, and this happens in science, of course. And what we saw was that these tau aggregates, as shown here, overlap with speckles. But we also saw that the speckle proteins, which are normally in the nucleus, these are normal cells. You can see the red protein is always in the nucleus. When you have tau aggregates, sometimes the protein moves from the nucleus to those tau aggregates. Right? And this is not unique to this particular protein. SRM2 does this the most of any other protein, but other proteins in nuclear speckles do this as well. All right, so the first question we asked ourselves, so that says that there's some interaction which causes proteins in the nucleus to move to these cytosolic aggregates of tau. The first thing we wanted to know, does this have anything to do with human disease? Because when you have a bizarre result that is new and different, the first thing you want to know is it important to the biology that you're studying? Because if it's, it could be some random thing that's just not that interesting. So in order to address this question, we went back and we communicated with Stan's lab, and Stan has access to a really terrific brain bank from individuals who got a number of different diseases and then passed away. And so, one group of patients that form tau aggregates are what are called people who have cortico-basal degeneration, where the basal ganglia region of the brain actually develops these tau aggregates. And so what we did is we obtained tissues from this brain bank and looked to see if the tau aggregates in those tissues contain this protein SRM2. And here's what one of those experiments looks like. And this staining was done by a talented woman, Felicia Oy, in Stan's group. So this is a patient down here who has cortical basal degeneration. And you can see this green here is staining for these tau aggregates. And you can see that they're all over the place in this individual. And there, if you look at an age-matched control, you don't see any tau aggregates, which is what you expect. Now, if we look at the SRM2 protein in the normal individual, that's present in the nucleus where it's supposed to be. But you can see here, it's now co-localizing with the tau aggregates. So just like we observed in the cancer cells in culture, in patients who've had these diseases, we see the same relocalization of proteins from the nucleus and from nuclear speckles to the tau aggregates. We didn't, of course, just look at one patient, we looked at many, and we see this result all the time. And it's not just in this particular disease, it's in all the diseases we've looked at. This is a disease, these are brains from individuals with Alzheimer's disease. Again, if we look at the age-matched controls, the protein is in the nucleus where it's supposed to be. In the Alzheimer's patient, we now see it out in the cytosol where the tau aggregates are forming. It's also true, these are some examples from frontal temporal lobe dementia patients. They're individuals who have particular mutations in tau, which drive this kind of disease. So this is a general property that we see in all of these tau aggregates, and we're really, because it's occurring in multiple different tau driven diseases in people, we think it's really important to understand this and then see what are the consequences of this, and is that something that drives the disease state? And that's something that we're working on. I'll tell you a little bit about as we go forward. All right, so what happens when a protein moves from the nucleus to these tau aggregates? And in particular, we're very interested in what happens to these nuclear speckles. And I'm going to show you a few things that change. So one thing that changes is that the protein is in the wrong place. So here we're looking at this SRM2 protein, and these two images are taken under the exact same exposure times and brightness. And you can see that here there's very bright nuclear speckles, and here they're much dimmer. And that's because about 50% of the protein has moved from the nucleus, where it's supposed to be, to this big tau aggregate, the cytosol. So you've removed proteins from the nucleus where they're supposed to be and moved them to the cytosol. And of course, that means this protein can no longer do its function as efficiently as it used to. And I want to point out that this redistribution of proteins is also seen in patients. And so if we go back to this cortical basal degeneration patient, here you can see in the normal individual, the SRM2 protein is in the nucleus, and in the disease state, the protein is now gone from the nucleus and is present in tau. And in fact, if we look at the reduction, you can see this is a big, it's over half the protein is removed from the nucleus and is now present in the cytosol. So you've removed key proteins from the nucleus and put them in a different place. The other thing that happens is that the organization changes within the nucleus. So typically in the nucleus, this is a normal cell, and we're staining for two different proteins, which are found in these nuclear speckles. And you can see that they largely overlap to form this structure called a nuclear speckle. But in a cell shown here, which has a tau aggregates in the speckles, you can see tau overlaps with this protein SRM2, but it pushes the red protein, which is a protein called SON, it doesn't really matter what it is for this, out to the edges of this assembly. So it's changed the organization of these structures. And we don't know what this means at a molecular scale, but what we do think is because organization allows function, if you change the organization, then you're going to change the function. And so because of that, we looked at function. And what people have hypothesized that speckles do is affect pre-mRNA splicing. And so we asked the question is, could the formation of these tau aggregates be changing pre-mRNA splicing? And the logic there is that tau aggregates form in nuclear speckles and alter them. The tau aggregates deplete the nucleus of, I showed you RNA binding proteins, but they also deplete some of the snRNAs that end up in those cytoplasmic aggregates as well, just like the proteins. And so maybe this process of splicing is now changed both because these RNAs and the proteins that are regulating this process are depleted from the nucleus. And so we took cells with and without tau aggregates, and we sequenced the RNA from them to see if splicing had changed. And this is what we actually see. So what we see is that cells that have tau aggregates have big changes in splicing. And so this is just showing you a plot where we're looking at the amount of introns which are retained in mRNAs as opposed to those that have been spliced. And you can see these are cells, these are mRNAs which have more intron in cells with tau aggregates. And you can see there's a whole bunch of them which accumulate many, many more introns because they've failed to splice appropriately. And so what this tells us is that at least in these tissue culture cells where we study tau aggregation, the presence of tau aggregates is sufficient to change and inhibit pre-mRNA splicing. How tau aggregates do that remains to be determined at the molecular level, but that's something that we're working on. But we think this is quite important because there are lots of alterations in splicing in individuals who have neurodegenerative diseases. And this comes from experiments where people have taken brains from people who have passed away either due to Alzheimer's or control brains, something perhaps someone who died in a car crash, and they sequence the RNA from the Alzheimer's patient and the control patient, and you see that there's tremendous numbers of splicing alterations in Alzheimer's. And this has actually suggested, has led some groups to suggest that maybe we should think of Alzheimer's and some of these other diseases as being driven by altered splicing per se. And so this is something that we're trying to understand in our simple model. Why does splicing change and how can we rescue that splicing? Because that might give us insights into what's going on in actually the patient condition. All right, so this has kind of led us to this kind of hypothesis for thinking about what's going on in some of these neurodegenerative diseases that are driven by tau aggregates, that these tau aggregates contain RNA, that RNA and tau aggregates helps to recruit RNA binding proteins into them so that they, and that when you sequester RNA binding proteins into tau aggregates, that changes where they're localized in the cell, changes their function, and can lead to changes in both nuclear speckle organization, but probably more importantly, RNA processing. And if you change RNA processing, you change all kinds of downstream gene effects because you're making a different set of proteins in the cell. I want to point out that this kind of model is very similar to how people are thinking about the disease progression in some forms of ALS. And so during ALS, which is another neurodegenerative disease, although it generally affects motor neurons instead of central nervous system neurons. And what happens there is that a protein called TDP-43, another protein, which is involved in pre-mRNA splicing, accumulates in cytoplasmic protein aggregates. And when it does that, it leads to changes in RNA processing, leading to misregulation of genes. And that's thought to be contributing to the driving of the disease state and eventually neuronal death. And that's similar to how we're thinking about tauopathies, where they sequester RNA binding proteins in the cytoplasm, lead to changes in RNA processing, which then can lead to cell neuronal death eventually. And so I think the general theme that we're proposing might be involved in both of these types of neurodegenerative diseases is that the cytoplasmic accumulation of these different RNA binding proteins really depletes their pool from the nucleus, leading to alterations in RNA processing, misregulation of genes, and then downstream consequences. All right, so I just want to wrap up here with a few speculative thoughts about, as you do science and make new discoveries, it's always important to think about where can that information go in terms of developing useful therapies. And I think one of the really important steps for us is determining some of the alterations that we see, particularly changes in splicing that we can understand the mechanism of. Do they actually occur in tauopathy patients? Most of our work is in mice models of disease or in cancer cell models of disease. And I think we need to get more information if this is actually occurring in the human pathological state. The other is that we want to understand how this might be transferable to understand therapy development. And I want to point out that there's one protein which, when you get rid of it, you can prevent tau toxicity. And so this is an experiment done in mice where if you have a mouse which actually develops a tauopathy disease, if you get rid of a protein called MSUT2, you can actually prevent those mice from getting sick. And this is really striking because MSUT2 is an RNA binding protein that normally is concentrated in these nuclear speckles. Now, nobody really understands why does getting rid of a nuclear speckle protein rescue the toxicity of tau pathology in a mouse. But because we're now suggesting that, look, tau aggregates actually form in nuclear speckles, and they alter the properties of nuclear speckles. Maybe if we can understand how MSUT2 alters nuclear speckles, both with and without tau aggregation, we could actually gain some insights. This is actually a big focus on my lab right now, trying to understand this process. And then we also think about, can we actually think about ways, if we can connect this to patients more directly, can we use this information for drug discovery approaches? And so, one possible screen that we're starting to set out to do as a research experiment is to see if we can find compounds that prevent taus, these green tau aggregates, from forming in nuclear speckles. And that those would be good tools to prevent this. And we could actually ask if it impacts the disease progression in mouse models. And if that does, that might even become useful as a therapy. And so we're also trying to understand what the molecular basis of these interactions is. The fact that tau aggregates form here says that there's some molecular environment there which promotes the formation of tau fibers in a particular place. And if we can understand that, I think we have the opportunity to think about ways to altering that process and develop the therapies. And so that's kind of some of the directions we're headed in my lab in the future. Now, I just want to end by thanking my lab. This is a picture of my group. It's a terrific group, and they've really done all the work, and our collaborators in Stan Prusner's lab. And I'd like to thank them all, and I'd be happy to take any questions from here.